All right, guys. So you guys have an outline. So it's just a brief, um, unfilled version of what I'm going over with you guys. Okay. And I have my notes just to guide me through. Okay. So I make sure I don't miss any points. And what you guys can do is you can just listen to this video along with me. Whether you listen, if you have ADHD, I recommend listening to it on 2x speed. I know that's available. I know that's an option. What you can do with me is just follow along and just get the information, and then you can listen to it again and take notes. That's a really good way to make information stick. You can do it however you want, but as somebody who studies human neurology and how we remember things and how we process things, that is a really good way to do it. Listen to it all the way through the first time, take your big notes, then listen to it again on 2x speed or whatever to really solidify the information. Okay. So right now, what we're going to do is a timeline. And I think it's really good because what it's going to do is going to give you an introduction to some terms. And then the more that you hear it as we're going through, the more it'll stick and the more you'll understand it. So if we can go through and have just the storyline of genetics itself, then you guys can really already have the vocabulary in your mind so that you can start making links and pathways to being able to remember it. Okay. So of course, most of us have heard of Mendel. If you have not, Mendel is basically the father of genetics. So let me tell you a little bit about Mendel. He was a monk and he was a gardener. So he was the gardener that unraveled all of this. And it was way before the 1860s that this was unraveled. He was working and he was doing experiments on pea plants, and he was noticing different traits and he was noticing different patterns. And that's something that I think is really important for you to understand is genetics equals patterns. Okay? It's just different patterns, and there's statistics, there's math problems, there's all sorts of things that allow us to look at these patterns, not just from like one generation to the next, but through expanses of generations, through expanses of populations. And it all started with Mendel. And this guy, he, like, wouldn't you like to have an adjective after your name? Like Mendelian genetics, right? So there is Mendelian genetics and there's non-Mendelian genetics. But what you can remember about Mendel is he started with pea plants. So thinking of this, and I like to start here because I took it from the perspective of Mendel, and I was like, okay, when Mendel started this, he knew none of the words like gene, genome, chromosome, allele. Like he's the one that made the rules of the game without even having the pieces of the board yet. Okay, so he started this in the 1800s. His research wasn't even uncovered until the 1860s, right? This is when his research was uncovered, but he had been doing these experiments for a long time beforehand. And so Mendel found traits in these pea plants. Traits would be tall, short, their peas were round versus wrinkled. Those are the traits that he had found. And then he found different laws. And I don't want to go over the laws right now because he has 4 laws, 4 postulates, so to say. But he found all these different rules. Like I said, the rules of the game without actually knowing the pieces of the game. Excuse me, the game. All right. Excuse me. All right. In 1902, this is Sutton and Boveri. What Sutton and Boveri found is by looking at Mendel's work, what they found was the actual chromosome. So you'll hear chromosome theory. This is the time that it was found. And what they discovered was that genes were carried on chromosomes. So Mendel found all these different patterns. Oh, sorry, I don't know if you guys can see me. The sun is changing. Let me shut this. Hopefully that helps. Yeah, okay. So where was I? Okay. So they found that genes were carried on chromosomes. And remember, Mendel didn't know anything about mitosis and meiosis. He didn't know about chromosomes. He didn't know about DNA. He just knew about patterns. So you guys can start there, remembering that he started with nothing, none of the information. That's how I got through it. Okay. But Sutton and Boveri, because of the advancements in microscopy, so microscopes were really developing around this time, they actually were able to prove chromosomes because of meiosis. Okay, so moving on, Morgan, our friend Morgan here, he started studying with fruit flies. And what he started to notice was there was a sex-linked inheritance. And we're going to be covering that in week 2, more about sex-linked inheritance. But what he found was that genes are arranged linearly on these chromosomes. So remembering, I'm going to see here. I'm going to do a darker color and then I'll do a lighter color. So remembering that our chromosomes, I'm going to draw it right here. They look like these little X's. They have that centromere in the middle. Okay? And then you have your centromere in the middle. Genes are found linearly, that's a hard word to say, on these chromosomes. And we're going to talk about the structure of it, but remembering that our DNA is that double helix, and that wasn't even found all the way up until 1953. But we're just thinking, here's our chromosomes. DNA wasn't even found until here. It wasn't even decoded until here, right? The genetic code, this is codons. So they were able to find those codons here, okay? So you can see that we're starting from not knowing anything. If that doesn't give you courage and encouragement and the possibility to believe in yourself, I don't know what will. But so chromosomes right here, what he found was that these genes are found in a linear sequence on the chromosomes. So one of the things that I do, you're going to hear terms throughout this, like genome. You're going to hear gene. Okay, and so what I want you to think of in chromosome, okay, so I want you to just have a little bit of an understanding. So let me just take you on my path. So genome, you can think of it as the library of instructions that make you, you. Okay, so we'll put this here, library. It's the actual library. The gene is the different recipes that make you up. The recipe of your eye color, the recipe of if you're tall or short, the recipe of your skin color, the recipe of how you utilize cholesterol. Those are all the different recipes, and they're all in different genes scattered along here. So the cholesterol gene could be here, and the eye color gene could be here. Now, that's not to say that that's where they really are. I'm giving you an example, okay? And then the chromosome is like the bookshelves, okay? The bookshelves that all these recipes are on, okay? The bookshelf. So you walk into the library and the bookshelf is holding all of these recipes, okay? The genome is your specific library, or a human genome, or your genome, whatever you want to say. The gene is the individual recipes, okay? And the chromosome is the bookshelf that they are sitting on, okay? So hopefully that helps clarify some things. So in 1928, there was a study done, and what they found, there was a mystery that happened. They're messing with bacteria, and what they found was a non-harmful bacteria suddenly became harmful. And they were wondering why that is. And so remembering that we didn't really know what DNA was. We just found chromosomes. We just found out what genes are. We're still looking in the cell and we see all sorts of different things under this microscope. What we're seeing is we are seeing genetic information, but we're also seeing what else is in the nucleus? A lot of proteins, a lot of ribosomes. So we're like, so what caused this? Was this switch over? Was this proteins, or was this something else? Was this the chromosomes? And so this is where the mystery began of us asking questions of how did this even change? Well, I call this this little experiment. The Avery and the Irish, because his name is MacLeod and McCarty. These are the Avery, MacLeod, and McCarty. They are the ones who did the experiment to show that it was not proteins, but it was in fact DNA. So it was DNA that created the change from nice to harmful. Okay, but this information was actually not really widely accepted until right here with this Hershey and Chase experiment. And I want to make sure I'm not missing anything. So what they found, Avery and the Irish, they found that DNA is genetic material. They still don't know what DNA is. Like they know DNA is genetic material, but they didn't know the structure of it. Remembering this happens with Watson and Crick. Okay, and that's pretty much most people in biology have heard of Watson and Crick. So Avery, MacLeod, McCarty, they discovered that DNA was what carried genetic material, but it wasn't accepted until Hershey and Chase, they proved it, that DNA was the genetic material and not proteins. And this was not accepted because if you look at it, proteins, what are they? They're strings of hundreds of amino acids. They're very, very, very complex. They actually thought that DNA was too simple to be what housed all of our information. At this point, what they knew, hopefully I didn't hit my microphone too hard, but what they knew was DNA had just a string of letters. And what are our letters? A, G, T, C. These two and these two go together, right? And they're like, there's no way, that's too easy. Whereas if you look at a string of amino acids, you have those 20 amino acids that make up tons and tons of different possibilities. So they thought DNA was too simple. Well, Hershey and Chase proved that it was a protein. They killed off different fats, carbs, proteins. They killed off genetic material. And it was only when genetic material was killed off that they found that it was able to transfer over. Okay. And then you had the big Watson and Crick, and I will give Franklin and hi, my cat just burst in here. And Franklin and Wilkins, the ladies involved, that did not get the credit back then. They found the actual structure of DNA. They found the structure of DNA and they found the replication, and they actually found that it stores information. So this little gene here is a storage of information. It's the information that makes the recipes of us, okay? Right? So in the 1960s, we actually found the genetic code. So we found codons, like the triplets. And we're going to talk about this when we talk about DNA, which is a little bit further down the road. Sorry, let me shut this. Okay, so let me see here. Genetic code was cracked, right? So we found triplet codons. So they come in 3. So our little letters will code for certain amino acids. And we're going to cover this, so don't panic. So different strings of these amino acids eventually make up proteins. So what in 1960, what was found was the central dogma of genetics. What they found was DNA goes to RNA, goes to protein. Okay? So that's a big important point. And if you can just remember that, you got it. That's good for physiology. That's good for a lot of different things right there. Just remembering that. Okay. So in the 1960s. So in the 1970s, that's when they started playing with it, right? So they found a enzyme within a bacteria that could splice genetic information and without destroying it. So they're called restriction enzymes. And they're basically like molecular scissors. So they could cut DNA and leave all the parts there that were needed to start replication and to insert it into DNA, into genetic material. So they found these little vectors that carry foreign DNA into a host cell. So these vectors were stable and they could take the DNA from one cell to another. So that's when we started playing with it. So we're starting here. Here's a Most people know about this, about Dolly the sheep, but this is when cloning started to happen. Okay, 1980s to 1990s. So they're really, really, really rapidly starting to decode this process. They're starting to see what genes are coding for what traits, right? Then in 2003, down here, we have the Human Genome Project. So that's where we figured out the human genome. We took it all out and we understand like this is where this lies and this is where, right, like what I was saying, the eyes lie here and this is what codes for different eye colors. Okay. Then we come into today, from 2000s until today. So we're starting to see next generation sequencing. So we're starting to be able to test for the next generation to see what's coming up, what's available. They're in the closet. What's available. We have CRISPR. This is gene editing. And we're going to be talking a little bit about is any of this process actually ethical? Should we be messing with things like this? Okay. And then we've also found medicines, therapies, and we're starting with synthetic biology. Again, it starts to get into, is this ethical to be doing this, to be messing around with DNA like this? So I wanted to give you one more way of thinking of this genome, gene, chromosome. You can think of your Netflix account. Okay. So your genome is your actual account. The episodes are your different genes. And then you get to pick then. Oh, okay. Let me say this. You have one series. That's your gene. Okay. So your Netflix account, your gene is your series. Okay. And then we'll start breaking it down even further. And we'll using words like exons. What are we doing here? Exons and introns. Okay. And so the language will get a little bit different. Now, we're not necessarily talking about mitosis and meiosis because you guys can watch the video, but I will cover it a little bit more. So the next step that we're going to do really quick, and I just thought this would be fun, a different way for you to experience cell biology, because you've been hearing it, you've been doing all of the things for learning the inner workings of a cell, and maybe you have it down and maybe you don't. But either way, the next step is we're going to just walk through the cell. And what I want you to do is set down all your pencils, everything that you have, get in a comfortable seat, sit up straight and start breathing. Sorry, I just heard a fall. Making sure everybody's okay. Okay. So sitting up straight, back nice and tall, and just closing your eyes and just breathing and using your imagination here because you've seen the inside of a cell. So we're just going to review it really quick in a non-panicked way because we're stepping into information that might be difficult. And I want to ground you back to, here's the thing. Mendel didn't know about all these different parts of the cell when he did all of this. He just started doing something and he started noticing something and he just followed it. Okay. And so I want to give you guys that same opportunity to just chill out. Don't worry about it. We're going to walk through the cell. I'm going to remind you which organelles do what, and the ones that are important for what we have going on right now. Okay. So closing your eyes, just taking some deep breaths. I want you to imagine yourself being really, really tiny. And whatever you imagine a cell to look like, you're walking up to the cell, and the cell is a big city. I really want you to have that image in your mind, remembering that a cell has a phospholipid bilayer. What that means is that your cell has like these little orbs, all these different balls that are floating around, and they're moving, and there's gaps in between them. So our phospholipid bilayer, you're seeing the phospholipids, the phosphate group. It's a little round glowing orb as you're entering this city. You can even think of it if you saw Atlantis. I don't know, I might be too young for you. Is that when the dome came over Atlantis to protect them. I like to think of that. But this dome is made up of a bunch of tiny little glowing balls, glowing orbs. And as you're walking this path to enter the cell, you can see it getting bigger and bigger, and you can see these orbs kind of moving around. And they have doorways, and there's windows into the cell, and there's different things anchoring it in place. Just get that image in your mind. Remembering to take a deep breath, because this material, we are going to learn it in a simple way just to get the foundation, not trying to get it perfect. As you're walking up to the cell, remembering that. You get to a door where you're just kind of pulled inside. So you have active diffusion. You're just pulling inside, or passive diffusion. You don't use any energy to get in here. So this is where we are going to be doing our work here for a little bit. We're going inside the cell to see how what's going on in here affects what's going on out here, what we look like, what we sound like, the diseases that we're susceptible to. Remembering that. Okay, so as we enter the cell, we're walking through this layer of orbs, and we can look around and we see these tiny little finger-like projections from both sides, that almost like they're coming at each other. Almost like they are coming at each other. And that's the little tails. Okay, and you can see on the other side, inside the cell, there's a layer of orbs as well. So that's our bilayer. There's 2 layers of this. Orbs on the outside, lipids, orbs on the inside. And what they're doing is they're protecting the cell. They're keeping this cell from getting dehydrated. They're protecting it from invaders. They're letting things in and out in a controlled way. They're keeping everything together. They're allowing for anchoring of structures. Okay. We're entering a eukaryotic cell. And if you remember, a eukaryotic cell has a membrane. It has membrane-bound organelles, and it has a nucleus. So you're entering the city of the cell. So it's glowing city as you enter it. You can see like a mitochondria on your left, and maybe you see a Golgi body on your right, and you see ribosomes floating around, little red balls floating around. Maybe you see some peroxisomes and some lysosomes glowing bright neon colors because of their functions. And then at the very, very end of the trail, you see the nucleus, big and important, kind of like you're walking up to the Wizard of Oz, to that Emerald City, that way down there at the other end. Okay. So as you start moving through the cell, you had your mitochondria, remembering that your mitochondria, it's whirring and busy and making noise. And you can see chambers stacked on top of each other, knowing that it's shooting energy through its layers. It's creating massive amounts of energy that are going to be used by this cell. Now I just want to make sure I have this right. You have your ribosomes that are floating around, and you have different types, and we'll go into those different types, but you have these red little balls of ribosomes. Some, they're like crystal looking, and they're stuck in the membranes, and some are floating around. And these ribosomes, they're the performers within the cell. They're making sure everything is working properly. They're making sure that there's proteins made. Okay, they are making things. Okay, we have our centriole. Our centriole is just like this glowing golden, I'm trying to think of how to explain it to you. It almost looks like a little tube that's shaped like a star. Long tube that's shaped like a star with a little piece coming out of it that's also shaped like a star. So you have these centrioles and these centromeres that are, they're stagehands. The centrioles are moving things around. This is inside your nucleus. So we're not there yet. Give me a moment. Sorry about that. So you're looking to your right, and you see the Golgi apparatus, and you can see it packaging up these proteins, preparing them to leave. And it looks like just big blobs of layers, just this big folded sheet. It's zigzagging and crossing, and you see proteins coming in, and then you see a big blobs going out, carrying the proteins out. It's packaging. It's like the post office. Okay. As you move further through the cell, you can see the endoplasmic reticulum. You can see some of this endoplasmic reticulum is studded with those ribosomes, and some is really smooth. Again, it's just folding networks, like you're passing through a city in Italy, just a whole bunch of zigzags and trails. And you can see on this smooth side that we're just getting big blobs of lipids. That's where it's packaging our lipids into. And then in the rough, you can see that we are creating proteins in here, that proteins are being created in this area. So, and they're being passed on to the Golgi apparatus to allow us to package those proteins. Looking up, you can see those peroxisomes and those lysosomes, they're floating around. They're glowing because they're carrying out wastes and they're taking out free radicals and things that could possibly damage you. And as you're walking along and you're heading to the nucleus, you can also see that this nucleus is membrane bound. And so you're going to pass through into this nucleus. And at the end of this nucleus, so you're inside this Emerald City, and you can see the nucleolus, which is where your ribosomes are all working really, really hard to get the DNA and translate it and transcribe it and all of those different things. We're going to go into those terms eventually. Okay, but you could see this little dark area where your genetic material, it's loose. We hear about chromosomes being tightly packed, but we're not replicating. We're not replicating. So we can see that this information is a little bit loosely packed so that these ribosomes and these enzymes and these proteins can get in there and they can make new. It's only whenever we start to divide and make new cells that this becomes really, really, really compact into that chromosome-like structure that you're used to seeing. Okay, so right now it's free floating. Then you see chromatin, which is this is that DNA. The scrolls are a little bit open. Like I said, the DNA is not tightly packed. It's a little bit open so that the workers, the ribosomes, the proteins can get in there and they can do what they need to do. Okay? You have the centriole. These are the stage hands. So when we do start to divide, they are moving the chromosomes to the right place. No, ma'am. They're probably in the closet. Okay. So as we're moving through the cell, we're seeing all of the different information. We're remembering that we're made up of trillions and trillions and trillions of these cells. And those that went before us, they didn't know all of this about the cells. They didn't know all the different parts of the cells. They just knew that they were seeing patterns. So as you're going through here, just try to get as many patterns as you can out of this process. Okay, you can slowly begin to come back if you were able to get that visual in your mind to travel through the cell. Okay. All right. So when we are stepping into this, one of the things that I want you to remember with mitosis and meiosis, I'm going to go ahead and erase this. So this is your time to screenshot if you don't have it, or just re-listen to it. Okay. We're going through here, remembering that a lot of this is going to be new. If I say something wrong, though, for some of you, for you pros and you experts, I shouldn't, but I kept it pretty basic. But if you pros and experts happen to be in this class and just need it for credit, I would love to get your feedback. I would love to get even deeper depth to this material. So don't feel afraid that if I say something wrong to correct me. Okay, I need you to exit the stage, please, because mommy is trying to get this done, okay? I also have 2 children that might interrupt me from here and now. Katie? She's in here with me, and I need her to step out, please. You don't need a whole bunch of stuff in here.
Hey, we're leaving. What are you doing?
If you know, you know. Okay? If you know, you know. Okay. So, real quick, you have a video over mitosis and meiosis. You can go watch that. But what we're going to do is we're going to cover just a little bit of information. I'm going to give you a brief overview. Okay? So, mitosis is basically photocopying. So, all of that genetic information that you had, it's basically copy and paste. Okay? Photocopying. The cells that this happens in is the cells that just need to divide and make new. So your skin cells, your liver cells, your hair cells. Those are the cells. They're called somatic cells. And we want exact replication because if we don't have exact replication, that's where we can get mutations. Okay? Meiosis, that is basically shuffling a playlist. So you got Spotify, playlist. And this is for gametes or sex cells. Okay? So when we look at that in chromosome 4, you have XX, which makes a female, and we're going to talk about this more, and XY, male. Okay? Now this is no like politically correctness. This is just genetics, okay? 2 X's make a female, X and a Y make a male, okay? We're not getting into politics here. This is just genetics. All right? Now, what is the purpose and the role of each different one? So with mitosis, purpose, we'll put this here, and we'll put mitosis, we'll put it in red. So the purpose, oh, I spelled purpose wrong. Here we go. Just know I don't spell well. So the purpose of mitosis is duplication. It's making more cells. Okay? The purpose of meiosis is shuffling, like I said, it's shuffling, but it is for genetic diversity. Genetic diversity. Okay? Let me flip this over. So the number of cells that we start with. So, oh wait, let's do number of divisions first. Number of divisions. Okay, number of divisions. These cells divide once. Okay, and then you end up with 2 cells. Okay, whereas here, these divide twice. So there's mitosis, and then there's meiosis 1 and meiosis 2. So they divide twice. Okay, the number of cells that we start with. We start with 1 here. We start with 2 here. Okay. Ending cells. I'll put cells. Okay. This one we end up with 2. This one we end up with 2 to 4. Okay, but we'll put 4. Okay. Chromosome number. It's important. I'm getting off on the color. Okay. Chromosome number. Okay. This is, we have 23 pairs or 46 chromosomes. So they have the full set. And this is for humans, okay? Whereas over here, we have 23, not pairs, but we just have 23 chromosomes. Remembering that These are our somatic cells, and these are our gametes. So our gametes is like sperm and oocytes or eggs. Okay. So we have half. We have half the amount because when we duplicate or when these two come together, it allows for 46. Okay. Hopefully that makes sense. Okay. Genetic identity, gamete, somatic cell. So we got that. We talked about the key roles, photocopying, shuffling the playlist. So that is our mitosis and meiosis in a nutshell. But like I said, I have a video for you guys to watch to get that information a little bit, like to have a visual. I'm somebody who needs to have a visual. I'm a very visual learner and kinesthetic. So that will help you remember. If you are watching something and drawing it, it will help you remember. Some of you have drawn mitosis and meiosis with me in the past. If you took physiology with me last year, you had to draw those. So there you go. You have it now for genetics. Okay. So when we're looking at our cell cycles, if we have a cell cycle that differs in duration, so if it takes longer, let's say for some of them will hang in limbo at different phases in there. And if you watch the video, you'll know. Okay. What would make the cells differ in duration of how long it takes them to divide? Well, sometimes we have the function. Depends on the function of the cell. Do we need this many this fast, or can we just have them prepared? The development, it takes longer. It takes longer for that to happen. Some of it has to do with actual nutrient availability. Do we have enough nutrients for this cell to divide? And the other thing is we're doing quality control. So we're making sure that the DNA is not damaged. There's not mutations. So we have little checkpoints along the way, as you saw in the videos. We have little checkpoints that make sure that all of these different cells meet certain criteria before we allow it to go on further. Okay. So in the video, it probably talked about homologous pairing. Okay. Homologous pairs in meiosis versus in mitosis. Meiosis, it happens because you are wanting diversity. We are mixing and then we're reducing the amount so that they can be in those sex cells. Okay. If we have too many of these homologous pairs, I guess so to say, we'll get non-disjunction. And that's when you get Down syndrome. You get too many or too little. You get certain chromosomal mutations. And that's where you start to end up with certain, I don't want to say disease, but chromosomal mutations. Yeah. So you have too many or too little pairs. Okay. Mitosis, we want it to be the exact same. And if we do not if we introduce homologous pairing in mitosis, what we'll do is we'll introduce errors into the system. Okay. So hopefully that made a lot of sense. Okay. Again, we're going to talk a little bit more about this in the future, but we're going to talk about sex cells. Okay. So we have spermatogenesis and we have oogenesis. So spermatogenesis, obviously, here, we can do this here. We can put location. We can put when it begins. We can put number of gametes. Okay. And we can put the timing. Okay. All right. So this is, we'll use those colors again. Making of sperm, spermatogenesis. Okay. Genesis meaning the beginning of, right? And oogenesis. Okay. So it's sperm versus the oocyte. Okay, this is how they begin, the beginning. So the location, obviously we know spermatogenesis happens in the testes. Oogenesis happens in the ovaries. Okay. The beginning, this begins puberty. This begins before birth. So we could even talk a little epigenetics here. Okay, epigenetics, which we'll talk about in the future. It's much more fun. Okay, the number of gametes, we have 4 functional. 4 functional, whereas here we have 1 functional, and we have about 2 to 3 little polar cells, polar bodies. Okay, and this happens again because we've split up all that genetic material, and we need to have 1 in this area because it will hold all that nutrition, all of the things that we need. They will funnel it all into 1 cell. Okay, all right, and this ends at this process ends at menopause, and this process pretty much keeps going forever. Okay, it slows down the older you get, but it still goes. Okay, all right, so we have talked about spermatogenesis versus the timing. Sorry, I forgot the timing. This one is continuous, and it's also rapid. Whereas this one, it's 1 egg per month, which we know that, right? 1 egg per month. So again, you were given all of your eggs were made before birth. You have 1 functional each month, 1 egg each month, and then you have the 2 to 3 polar bodies. Okay, and we'll dive into that a little bit more when we talk about the sex-linked genetics. Okay. Again, if you have any questions, it's good to write them down right now. I might answer them in a little bit by the material, or you can email it to me, but you don't want to forget the question. Okay, and mistakes as well. If I made a mistake, email it to me. I want to know. All right. So going back, now we are going to go back to Mendel's time. So I'm going to put Mendel here so you guys remember. Whoop, Mendel. Okay, we are going to be talking about Mendel and his 4 postulates. Okay, so I'm going to write down the 4 postulates right now, remembering that he was a gardener, right? Technically, they'll say he was a monk, but he was a gardener at the monastery. Okay, he used pea plants. Okay, he used these pea plants to come up with these 4 postulates. Okay, what are the 4 postulates? Well, let's talk about it. The first one is he found these unit, did I spell that right? Yep, factors. Okay, that come in pairs. Sounds familiar. Okay, he also has dominance and recessive. Right? Remove that. He has the law of segregation. And last but not least, law of independent assortments. And you guys get what all of this means, I'm sure. Right? That's all I needed to say, right? Just that reminder. I'm just kidding. Just kidding. Unit factors in pairs. So what he noticed was that you get 2 copies and you get 1 from each parent. So 2 copies, one from each parent. Ooh. Okay. All right. If we look at this, so this is in the Mendelian view. If we look at this from our modern view, what this is, so let's look at it like tall, short. Okay. Round, seeds, wrinkled seeds. Okay. All right. Those are the different things. If we look at this in our modern view, this is genes. Genes. Remember, this is our different recipes that make us up. There's a recipe for tall. There's a recipe for short. Those are different alleles. Introduce that term. Okay. All right. These are specific DNA sequences. So DNA sequences at fixed loci. So remember when I drew that chromosome? Okay. This is a loci. This is a loci or locus. So these are loci. This is a locus. Okay, so these are different locations on the chromosome. So DNA, so the genes are DNA sequences at fixed loci on homologous chromosomes. So this is a homologous chromosome right here. So it's when you're looking at our 46 pairs, right? 46 pairs of homologous chromosomes. This is one. Now you might find one that's like a homologous chromosome is the ones that are matching. So if you were to look at a picture of all of them together, you can see that they kind of get smaller. Some of them are really short up here. Some of them are really long. But what they found was that these are homologous. So they're at specific locations on specific chromosomes. The trait for eye color is on chromosome 1. You know what I'm saying? The pair of chromosome 1. So that's what they found. Unit factors are DNA sequences found on specific loci within a homologous chromosome, a matching pair of sister chromatids. Again, we can talk about that a little bit more. Okay. Dominance versus recessive. So he found that 2 different unit factors are present. One may mask the other. So the tall usually match short. So they write this for the Punnett square. You've probably seen this. Tall and short, right? The tall is dominant. So when it goes, here we say we cross 2 talls, 2 shorts, right? T, T, that's dominant. And we look over here, T, T, that's this tall is going to be dominant. T, T, this tall is going to be dominant. T, T, that one's going to be short, right? There's your Punnett square introduction, okay? The modern view, what they have found is that it is not necessarily whether one is dominant or recessive, but what they have found is the functional or non-functional proteins associated with that gene. So we'll put that here. Functional versus non-functional proteins. Okay? And that determines whether or not it's dominant or recessive. And that has a lot to do with environment, which they didn't think it had anything to do with environment, but we're starting to see the environment before birth, before the eggs were even made, and the environment that the genes are in has a massive, massive impact on whether that gene is expressed. So the genotype, I'll say this, the genotype is your mixture of alleles. So genotype is how your alleles look, your mixture of, so this is your genotype for here. This is your genotype for here, right? Your phenotype is the actual trait itself that shows up. So genotype is what your genes look like, basically. Your alleles look like as they're passed on. Tall and short is your phenotype. That is the physically expressed trait. Okay, hopefully I explained that good. Okay, law of segregation, right? So we have this here. So we have a coin flip. That's the law of segregation. So I'm going to put that right here. So a coin flip. And you can see that up here, because we have two copies, heads, tails, we can look at it like a coin flip. They have nothing to do with each other per the trait. It's just, ding, which one am I going to get, head or tail? Because you have two options, okay? I don't know why I wrote that. But like I said, we have that separation. When we separate in meiosis, you get one copy from each parent. So it's a coin flip, okay? And then the law of independent assortment. This one has to do with how different traits behave relative to each other. And we're following traits at one time. You can follow two traits at one time, okay? So are we going to get this and this, or are we going to get this or this? And we'll talk more about that as well. Now, when we look at this, he did different crossing. So he did tall with short, okay? So that's what we have here. He did round with wrinkled. Okay, we could say that, the same thing. Now, when he started, this is a monohybrid cross, right? Monohybrid. So he's just crossing these two traits, okay? And then what he found, and you guys can look this up in your book. I'm not going to draw a super long, detailed thing. You can either use the Punnett square or you can use the branching method, whichever works. What I found is the Punnett square works really well for the mono. And then once you start getting into more, the Punnett square gets a little chaotic. So I just do, for the first layer, I do mono. So if it's a dihybrid, I'll do both traits as a monohybrid, and then I'll start to cross them. And I'll show you an example of that here in a minute. Okay. But here's the thing. He found all three of these laws using monohybrid crosses. But this law has to have two traits. Two traits. We have to follow two traits to know that two traits will happen independent of each other. Remembering that he didn't know what a gene was. He didn't know what a chromosome was. So he had to look at, okay, what if I cross all of these together? You know, what if I cross a tall, short, round, and wrinkled? Okay, so that's a dihybrid. And then he was talking about three, so the color of the seeds. That's a trihybrid cross, okay? And so that's where he found this, within the law of independent assortment, he found probability, okay? The reason that Mendel was so successful is because, one, his choice of organism, was these plants, right? And what this is called is a model organism. So today we use model organisms. We use bacteria like E. coli. We use viruses. We use mice. We use things that can reproduce and make a lot at one time. And they have short lifespans. And here I have a whole, let me just put this, let me come back here and I'll talk about them. So they're easy to grow. Model organisms, that's what we're talking about. Model organisms are easy to grow. They have short life cycles. They have many offspring, and their genes are pretty straightforward, okay? So that is the reason, one of the big reasons that Mendel was so successful, okay? The way that he designed his experiment, he focused on single traits, and then he expanded. He took a very quantitative approach. He had clear framework, and then he was very persistent within this. This was like I said, his research was 1860, but he had been doing it for years before. So he was persistent in looking at all of this, all of these patterns. You know, he was basically just having fun with flowers, y'all, which flowers are a really fun way to learn genetics, by the way. Okay, so let's look at this. Monohybrid is like flipping one coin. Okay, that's tall, short, head, tails. Dihybrid is flipping two coins at one time. Tall, short, round, wrinkled. Okay, trihybrid, tall, short, round, wrinkled, like flipping three seeds or three coins at one time. Yellow, green. Okay. The reason for Punnett squares and branch diagrams is really just to give you all possible outcomes. Okay, so we're just looking at the possible outcomes. So if that's exciting to you, the possibility of patterns, welcome to genetics. Okay, so when we're looking at the laws, I'm going to erase this. Looking at independent assortment wasn't found until he started using dihybrid and trihybrid crosses, because the independent part, like these different traits, are independent of each other. Okay, they'll happen independent, they don't affect each other. Okay, and we're going to look at exceptions to all of that here in a little bit, but I want to, hopefully you guys have this. I'm going to erase it now. We are almost done. Hang in there with me, guys. Okay. Looking at these laws of probability, right? So he's a pattern and he likes the patterns, and then we're going to look at probability. Possible. What's the probability that this could happen? So laws of probability. So we already talked about one, law of segregation. That was our single coin toss. So 1/2, right? There's a half a chance you'll get this one and a half a chance you'll get the other one. Okay. I have this all written, all sorts of everywhere, right? So these genetic ratios are expressed as possibilities or probabilities. So the law of segregation is our coin flip. So alleles separate randomly. So these are our different traits, tall, short, that allele, they separate randomly. So your probability is 1/2. So you got a half a chance of getting head. And this works better when you have more flips. So what he found was there was a 3-to-1. So you saw with that Punnett square, there was a 3-to-1 ratio of getting tall to short with that Punnett square that I just had right under there. That was a 3-to-1 ratio. And when you start getting bigger, it's like 9, 3, 3, 3, 1. Okay, you start getting into different ratios. Okay, so these are all the probabilities. Law of segregation. You have a coin toss. Half a chance you're going to get heads, half a chance you're going to get tails. Next, we have product rule. And basically what you can remember is this is and. So this is adding math. I know. Adding math into genetics. Okay. We have two parents that this is what their genome looks like, or their genotyping is. Okay. Oh, I have R times. So when it's and, you're multiplying. Okay. So we have a half a chance here and a half a chance here. And so the probability is 1/4. Okay. So we have these two. These are our parents. We could say dad, father, mother. Okay. What's the chance we're going to get RR? Okay. R and R. So what is the chance that we're going to get R and R? Half a chance equals 1/4. So you're going to multiply. So you're flipping two coins and you're asking the chance of getting heads and heads. Okay, so we could put that here. Head and head. Okay, the and is the key word. What's the chance we're going to get that? We want to get both things at once. Okay, then we can come look over here, or we'll just do this right here. Sum rule. Sum rule. This is all in your book, guys. And isn't that a nightmare? Wouldn't you hate to read this? Okay. This is or. Okay, this is our or. So this is like rolling a dice. So I know that it did coins up there, but this is like rolling a dice. So we roll a dice. What's the chance we're going to get a 2 or a 4 when rolling a dice? Okay, so we can see this or right here. So there's 6 numbers on a dice. So we have 1 6 and 1 6, a chance that we're going to get either of those. Okay, so what we're going to do is we are going to add. So when it's an and, you multiply. When it's an or, you add. Okay, so 2 sixths, so 1/3, right? 1/3 of a chance. What's the chance of a 2 or a 4? So we have an R dad plus an R, or an R dad plus an R mom, 1/4. Okay, R dad plus R mom equals 1/4. So we have 1/4 plus 1/4 equals 1/2. Okay, one or the other. Okay, we want to get this RR. What's the possibility? Okay, hopefully that makes sense. Why it matters is these are more complex crosses, right? So once we start getting into more complex crosses, that Punnett square and that branch chain, or that branch chain, I almost said branch chain amino acid, that branching, that can get pretty big. So this is a way of looking at it that's a little bit more simple. It's layering it on. Okay, it's layers of probability. Okay, so I want to show you what this looks like really, really quickly of the tri-hybrid, how I did this. And it's kind of messy, but at the same time, it's so much easier. Okay, so he had a tri-hybrid cross. Okay, and what I did was I mixed and matched all. I'm going to give you a good example, and let's hope that I can do it. Let's hope I remember how to do it. So you have, let me say this, round and wrinkled seeds. So we will say round and wrinkled. So we're going to do a Punnett square where we have our round and our round. Yep. Oh, wait. We don't need both of those. Round and wrinkled. Okay? So we're going to get RR, 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 RR. So here's your 3-to-1 ratio, right? 3-to-1 ratio. Okay? Then we have yellow versus green. Same thing here. Yellow. I'm going to do a little G because it looks, my Y's kind of look similar. Okay? Y, 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 G. And it really doesn't matter what letters you use, by the way. Again, 3-to-1. So the green, here, let me put this right here. Y, G. Okay? 3-to-1. So you can see that this is the recessive. Recessive. Dominant. The dominant, because this is the dominant. You have, oh, no. This is recessive. This is dominant. 2 recessive. It takes 2 recessive for that trait to show up, for that phenotype to show up. 2 recessive for that phenotype to show up. Let's do one more tri-hybrid cross. Tall, short. This is with pea plants. Remember, okay, we have tall, we have short. T, S, T, S. You're crossing tall with short. And then we're going to cross their offspring. Okay? Tall, 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 short, tall, short, tall, or short, short. Okay, so again, 3-to-1 ratio. These are our ratios. Now, let's say we have our 3/4 round, right? Because we have 1, 2, 3 out of 4. 3/4 round. And we're going to cross it with the yellow and the green, right? So we have a 3/4 round and we have a 1/4 round. 1/4 wrinkled. Okay. We're going to make like a little Y, and we're going to come here and we're going to do our 3/4 yellow, 1/4 green, sorry. And we're going to do the same thing here. 3/4 yellow, 1/4 green. Okay. Then we are going to cross this over here. So now we're going to take this one and we have a lot more space. We have 3/4 tall. We have 1/4 short. We have 3/4 tall. We have 1/4 short. We have 3/4. I know it gets a little redundant, but 3/4 tall, 1/4 short, 3/4 tall to 1/4 short. So then I'm not going to do all of this math, but we're going to come across and we're going to multiply here, here, and here. So we have 27/64, right? Then we're going to go here, 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 multiply. So that is 3/64. Okay. Again, I'm just going to take it through one more time. We're going to multiply here, here. Oh, wait. Yep. Here, here, here. Well, we got to go here, here, here. Okay. So that is, again, that's going to be our, that's 6/64. Then we're going to go here, here. You see how you can follow it through? That's going to be our 3/64. And of course we need to reduce those, and I'm not going to do it for all the way through. But this is so that we can determine our ratios a little bit easier. So that's a tri-hybrid cross. And I use the Punnett square and then I take it to the branching to figure that math out. Hopefully that helps you. That's our law of probabilities. That's multiple crosses, that's layering, things like that. Okay, so the purpose of this is the question. Is it about one allele splitting into two gametes? If the answer, here, let me erase this because I think this is really good. Again, looking at our laws of probability, the law of segregation versus the product rule versus the sum rule. Okay, what do we need to know? Like, what's the information? Why is this relevant? That's the question I asked myself when I was learning this and I was getting frustrated with it. What is the purpose? Okay, is it about getting one allele splitting into two gametes? Ask yourself that question. If the answer is yes, law of segregation. I'm not going to write that whole word out. If the answer is no, the next question that you need to ask yourself, does it ask for both things? So head and head, head and head. If the answer is yes, product law. If the answer is no, sum law. And that's your adding, this one here multiplying. This is your and, see, and this is your or. Okay, hopefully that really helps clear that up. All right, almost done, guys. We're almost done. I believe. Okay, so we're going to cover this really quick and then we'll take a break. All right. Okay. So probability plus statistics meets genetics. So we just talked about math meeting genetics with probabilities. So now we're talking about probabilities and statistics. So the statistics is the chi-square. Okay, so we have the null hypothesis, right? So when I wrote down the ratio 3-to-1, that's our null hypothesis. This is what we expect. Oop. Yeah, I did just misspell that. Expect to get. Okay, but what if we don't get that? So this would be 75% are tall, 25% are short. Sorry, that looks like a 3. That's what we expect. But what happens if it's like 68-32, right? That's where this chi-square comes into play. So this is what we actually observed. Okay, happen. Now what the chi-square does is it's an equation that allows us to see, are these just little deviations or is something wrong? Is something off? Is there something fishy going on here? Okay, so let's apply this. If we're testing a disease gene, if we're testing to see if a disease gene is inherited in a dominant versus recessive pattern, that's when you would utilize this null hypothesis versus the chi-square or the chi-square, however you want to say it. The null hypothesis is that there's no real difference between observed data and what you expect. So there's no real true difference. So this chi-square formula will allow you to tell the difference. Now, I'm not going to make you use that formula, but at least you know. At least you know, okay? I will cover the last little bit of this in a separate video. And again, if you have any questions in this first video, reach out, let me know. If there was something that I missed that you think is really important, reach out, let me know. I'm all open and I'm all ears. Okay, thank you guys.